Hello viewers, I welcome you back to my YouTube channel. Today we'll be talking about wind signaling. We'll try to understand the detailed mechanism of wind signaling. What are the different components involved in wind signaling and why this is important to study wind signaling. How this affect different physiological aspects of our body. So these are some of the important aspects that we are going to discuss in today's class. So, as we have discussed earlier also in the previous lecture, cell signaling, that there is cell 1 and we have another cell, cell 2. Now, this cell 1 releases a ligand. Ligand is a chemical molecule through which one cell try to talk to each other, communicate to each other. Now, this ligand goes and binds to the receptor which is expressed on cell 2, and then the interaction of this ligand and receptor is going to have some effect on cell 2. Now, this ligand might be of different types, and then this receptor can also be of different types. Like let us say, if we talk of some ligand like wind, this is one of the ligand which is involved in wind signaling, and that is what we are going to discuss today in today's class. Another ligand can be, let us say, TGF-beta. This wind ligand goes and binds to a receptor which we call frizzled. In short form, we write it like FZD. And TGF-beta goes and binds to a receptor called TGF-beta receptor or TGF-beta R. So, in today's class, we are going to discuss how this wind ligand binds to frizzled. So this ligand will be frizzled. This ligand will be wind, and this receptor will be frizzled. So today we'll try to understand when this wind ligand binds to this frizzled receptor, what are the molecular changes happening inside this cell. Another thing which is to notice is, in most of the time when you are studying in your textbook, generally we don't discuss much about this cell 1. What I mean is the cell which is producing the ligand or secreting the ligand. We do not discuss much about this cell 1. Our most of the focus is on this cell 2. So in each of the cell signaling topics that we discuss, generally we start with this ligand binding to the receptor, and then this ligand and receptor interaction has some physiological effect, metabolic effect on this cell. So all the topics generally starts with this ligand and receptor interaction, and then what are the molecular changes which are happening inside this cell, and what are the effects which is being caused by this ligand and receptor interaction. So in today's class we will try to understand how this wind ligand affect the target cell by interacting with frizzled receptor. So let us look at the diagram to understand this process clearly. So here we have two cases. This is case 1 and this one is case 2. So left panel of this diagram shows a case where there is no wind off. So wind off means there is no wind ligand is not present. This case is talking about when wind ligand is present. So let us try to understand case 1 first, when there is no wind ligand. So if this is the cell, this cell has one receptor here, and this is ligand which is going to interact. Now when there is no ligand, there is no Wnt in this case. In that case, what is going to happen is, in the cytoplasm of this cell there is a complex called destruction complex. This destruction complex consists of these molecules, which you can clearly see in this diagram. So this complex named destruction complex. This destruction complex consists of APC, GSK3 beta, exine, CK1 and beta TRCP. Now you can ask a question why we call it as destruction complex. The very simple reason being it called as destruction complex is because this complex try to destroy or destruct this beta-Catenin. In some or other way, whatever the components you are seeing here in the destruction complex, those destruction complex helps in destruction of this beta-Catenin. And since these components are involved in destruction of this beta-Catenin, that's why we call this complex as destruction complex. This beta-Catenin is one of the main player in this Wnt signaling pathway. So when this destruction complex destroys this beta-Catenin, there is no free beta-Catenin, so there is no beta-Catenin here inside the nucleus. So this one is nucleus. This one is cytoplasm. So this looks very simple. When there is no Wnt ligand, this destruction complex, which consists of GSK3 beta, exine, APC, CK1 and beta TRCP, is going to destroy this beta-Catenin. There is no free beta-Catenin to go into the nucleus. So if there is no beta-Catenin in the nucleus, this target gene will not be activated. This target gene will remain suppressed. Now how this beta-Catenin is destroyed? This beta-Catenin is destroyed by GSK3 beta component of the destruction complex. So GSK3 beta is glycogen synthase kinase 3 beta, and since this is a kinase, this has the capability to add phosphate group to the beta-Catenin. Now this phosphorylated beta-Catenin undergoes proteasomal degradation. There is one structure inside the cell called proteasome, which is involved in destroying the proteins. So once beta-Catenin is phosphorylated by this GSK3 beta, this beta-Catenin is destroyed by this proteasome. Another component of destruction complex which helps in destruction of this beta-Catenin is beta TRCP. This beta TRCP actually adds ubiquitin group on this beta-Catenin, which is shown here as UB. So UB is ubiquitin. So the destruction of beta-Catenin is mediated by two components. One is GSK3 beta of the destruction complex. What this GSK3 beta does, this GSK3 beta adds phosphate group on the beta-Catenin. Another component of the destruction complex, beta TRCP, this is going to add ubiquitin group on this beta-Catenin, and because of this phosphorylation of beta-Catenin and ubiquitination of this beta-catenin, this beta-catenin is going to get destroyed. Now if there is no beta-catenin, then there will be presence of no beta-catenin in the nucleus, and there is no beta-catenin, that's why there will be no activation of this target gene. Now let us try to understand the second scenario when there is the presence of this Wnt ligand. So when this Wnt ligand is present, this Wnt ligand is going to bind to this Frizzle receptor and LRP56 receptor or co-receptor. LRP56 is called co-receptor because it is associated with this Frizzle. Co is derived from the term cone, and you might be knowing this cone means together. So since this LRP56 is associated with this Frizzle receptor on the plasma membrane, that's why we also call it co-receptor. So this Wnt ligand bind to LRP56 co-receptor and Frizzle receptor, because of which this receptor gets activated. Now when this Wnt ligand binds to this Frizzle and LRP56 receptor, this receptor is going to recruit this disheveled protein to the Frizzle receptor, and this disheveled protein, which is recruited to the plasma membrane because of this Wnt and Frizzle interaction, this disheveled protein gets polymerized. Now because of this polymerization of this disheveled protein, this disheveled protein is going to disassemble this destruction complex. So there is no destruction complex here now. And since there is no destruction complex, so this beta-catenin will not be destroyed, and this beta-catenin is free to enter into the nucleus. Once this beta-catenin enter the nucleus, this is going to interact with TCF and LEF complex, and it is going to activate the target protein. One more thing which we can understand here is, when there is no beta-catenin, this GROCO and TCF-LEF acts as a repressive complex. When we say repressive complex, it means this GROCO and TCF-LEF is going to suppress the expression of this target gene. But when this beta-catenin associates with TCF-LEF, GROCO is released from this target gene, and beta-catenin TCF-LEF acts as activator of this target protein. So TCF-LEF can be considered as having dual role. So TCF-LEF associated with GROCO, it is a suppressor of target gene, but TCF-LEF associated with beta-catenin, this is the activator of Wnt target gene. So we can easily understand from this diagram now that presence of Wnt signaling disassembles the destruction complex, and because this destruction complex is disassembled, beta-catenin is free to go into the nucleus and activate the target genes. Now this is another diagram from which we will try to understand this Wnt signaling pathway. As you can see here, there is no Wnt ligand present. In that case, this destruction complex is going to remain active, and this destruction complex is going to phosphorylate this beta-catenin, and this beta-catenin will be destroyed. So in absence of Wnt ligand, there is no beta-catenin, and hence this target protein is suppressed. Target gene is suppressed. Now when this Wnt ligand is present, this Wnt ligand is going to bind to this LRP56 and this Frizzle receptor. Because of interaction of this Wnt ligand with LPR564 receptor and Frizzle receptor, this disheveled protein is recruited at the plasma membrane, and this activated disheveled protein is going to disassemble the destruction complex. Now if disassemble destruction complex is present, like this destruction complex is disassembled, in that case beta-catenin is free to go into the nucleus, and beta-catenin along with the TCF LEF is going to activate the target gene. So same concept which we tried to understand in the previous diagram is again explained here to make it more clear. I hope you can easily understand this now. Now whatever we had discussed so far, that is known as canonical Wnt signaling. But here we are going to talk about non-canonical Wnt signaling. So it means Wnt signaling can be of two types. One is canonical, another one is non-canonical. So whatever we discussed previously was canonical Wnt signaling. What is the difference? The major difference is that canonical Wnt signaling involves beta-catenin. So beta-catenin has central role to play in case of canonical Wnt signaling. But in case of non-canonical Wnt signaling, which we are going to discuss here, you don't find beta-catenin here. So the basic difference between the canonical and non-canonical signaling is presence or absence of beta-catenin. Now in case of this non-canonical Wnt signaling, Wnt ligand binds to the Frizzle receptor. Frizzle receptor recruits this disheveled protein. Now this recruitment of disheveled protein actually suppresses this protein DAM1. DAM1 is actually the inhibitor of this row. But when this DAM is suppressed by this disheveled protein, now this row is free. So dishevel mediates the de-inhibition of row by DAM1. Since DAM1 is the suppressor of this row or inhibitor of this row, and DSH is the inhibitor of this DAM1, so actually this disheveled acts as activator of this row. So in presence of activated disheveled, this row protein is activated. This disheveled protein also activates another protein called RAC. Now this RAC protein is going to activate this GNK. Here this row protein will activate ROC protein, and this ROC and GNK together is going to activate the actin polymerization. This actin polymerization leads to gastrulation. So gastrulation you might have studied in embryonic development. So from this diagram, this also becomes very clear that this non-canonical Wnt planar cell polarity pathway has very important role in embryonic development. Now this one is another non-canonical wind signaling pathway, which is known as wind Ca+2 pathway. From the name itself, this is very clear. We call it wind Ca+2 pathway because it is involved in regulation of calcium ions. When this Wnt ligand binds to the Friedel receptor, this actually activates G protein mediated phospholipase C molecule. The activated phospholipase C is going to activate IP3 molecule, and IP3 molecule is going to mediate the release of calcium ion. The way it acts is, the calcium molecule are generally stored in vascular structures inside the cytoplasm. Now these vesicular structures have IP3 receptor. This IP3 is going to bind to this IP3 receptor and it will mediate the release of calcium outside this vesicular structure, and that is how it works. So Wnt ligand will bind to Friedel receptor. This activates disabled protein, and there is a G protein mediated activation of phospholipase C. Phospholipase C is going to activate IP3 molecule, and IP3 molecule will mediate the release of calcium ion into the cytoplasm. Now this release of calcium ion in the cytoplasm is going to mediate many pathways which are like calcium regulated, like this is going to activate this calcineurin protein, this is going to activate this CAMK2, and this might also activate this PKC. So this increase in calcium might have multiple effects. It can affect the multiple pathways which can finally lead to ventral fat determination or tissue separation. Again, this ventral fat determination and tissue separation, you can easily make out from here that these are the processes involved in embryonic development. So Wnt calcium pathway and planar cell polarity pathway, which are two different types of non-canonical signaling, these are mainly involved in embryonic development. And canonical Wnt signaling are well known to be involved in different types of diseases and colon cell development and all. Now in this cartoon you can clearly see the difference of in different types of Wnt signaling pathway. So this is Wnt canonical pathway and these two are Wnt non-canonical pathway. So when Wnt ligand binds to this Friedel receptor, in case of canonical pathway, this will lead to transition of Wnt from the cytoplasm into the nucleus and activation of the Wnt target genes. In case of non-canonical pathway, this might be Wnt Ca+2 subtype, or this might be planar cell polarity subtype. In case of Wnt Ca+2 pathway, this is going to affect the cell adhesion and cell movement. In case of planar cell polarity pathway, this is going to affect the cytoskeleton structure, or it might induce the cytoskeletal rearrangement inside the cell. So based on what kind of Wnt ligand is binding to the receptor, it might have multiple effects on the target cells. Now we have seen the different types of Wnt signaling pathway. We have discussed about canonical Wnt signaling pathway. We have discussed about non-canonical Wnt signaling pathway. Non-canonical Wnt signaling pathway consists of planar cell porosity pathway and Wnt+2 pathway. Now, whatever we have discussed so far, let us have a quick recap on that. In canonical Wnt signaling pathway, we talked about destruction complex. Now, what are these destruction complex? These destruction complex are actually proteins which consist of axin, GSK3 beta, CK1 and beta TRCPs. Now, beta-Catenin is phosphorylated by GSK3 beta as we have discussed and ubiquitinated by beta TRCP, and this is targeted for proteasomal degradation. Then we also discussed about repressive complex, which is TCF-LEF associated with GROCO, and this TCF-LEF GROCO component is going to recruit SDAC, which suppresses the Wnt target gene. Then we discussed Frizzle can attach to disable in the existence of Wnt ligand. We also discussed phosphorylation of LRP56 via CK1 alpha and GSK3 beta agars, allowing LRP56 to interact with destruction complex. Canonical Wnt ligand consists of Wnt3A and Wnt1, and it binds to Frizzled and LRP co-receptor. We also discussed that LRP receptors are phosphorylated by CK1 alpha and GSK3 beta. Phosphorylation of this LRP by CK1 alpha and GSK3 beta leads to recruitment of disabled protein on the plasma membrane, and on recruitment of disabled protein on the plasma membrane, they polymerize and are activated. The activated polymerized disabled protein is going to sequester the destruction complex in the multivascular bodies, and this will lead to the free beta-Catenin to go into the nucleus. Now, stabilization and accumulation of beta-Catenin leads to translocation of this beta-Catenin into the nucleus, and inside the nucleus this beta-Catenin forms active complex with LEF. This LEF beta-Catenin complex is also going to recruit other protein components on the target gene such as CBP-P300, BRG1, BLC9 and PICO. All these are going to form a transcription complex on the target gene, which will lead to activation of the target gene. In the non-canonical Wnt signaling pathway that we discussed, the ligands which are responsible in that non-canonical signaling are Wnt4, Wnt5a and Wnt11. Non-canonical Wnt signaling is defined by beta-Catenin independent mechanism. So we have already discussed beta-Catenin is the major difference between canonical and non-canonical pathway. Beta-Catenin is primarily present in canonical Wnt signaling pathway, and there is no presence of beta-Catenin in non-canonical pathway, and that's the reason non-canonical Wnt signaling pathway is also known as beta-Catenin independent pathway. During Wnt PCP signaling that we discussed, Wnt ligands binds to the ROR Frizzle receptor complex to recruit and activate disabled. Disabled protein binds to a small GTPase Rho by de-inhibition of the DAM1 protein we have seen in that previous diagram. The small GTPase RAC1 and Rho together triggers ROC and JNK proteins. Activation of this ROC protein and JNK protein leads to rearrangement of the cytoskeleton and transcriptional response, for example by ATF2. Next to disable, there is another receptor called Wangle. This is also a key member of the Wind-CP PCP signaling, and this is activated by phosphorylation by Wnt5A dependent minor. Wnt+2 signaling is initiated by G protein triggered phospholipase C, leading to intracellular calcium flux and downstream calcium dependent cytoskeleton error and or transcriptional responses. Wind PCP ligands, Wnt5A, Wnt7 and Wnt11, these attach to the Frizzle receptor, and it leads to activation of a small GTPase Rho and RAC family, and JNK mediated by disable exert the downstream effectors that guide cytoskeleton structure and direct cell polarization within the epithelial sheet plane via regulation of the target gene transcription. The small GTPase, RAC and Rho collectively activates kinases like Rho and JNK that contribute to downstream C-zone and terminal kinase expression and transcription factor activation. Now, so we discussed so far two types of Wnt signaling pathway, canonical Wnt signaling pathway and non-canonical signaling pathway. Now, why this is important? Why we should study this Wnt signaling pathway? So this slide highlights you the different aspects where this Wnt signaling is involved. Wnt signaling is involved in the development of different types of cancers. Dysregulated Wnt signaling might cause colorectal cancer, it might cause breast cancer, it might be involved in prostate cancer, pancreatic cancer, melanoma, and other types of cancer. So this Wnt signaling, especially canonical Wnt signaling pathway, are very important when we talk of cancer. This Wnt signaling is also known to control the cell proliferation and differentiation of the cells. Wnt signaling is involved in maintenance of stem cells and spheroids. As we have discussed about non-canonical Wnt signaling pathway, we have discussed there that it is involved in embryonic development. And if we talk of embryonic development, this Wnt signaling is mainly involved in neural crest induction and specification, dorsoventral and antero-posterior body axis formation in the embryo, cell migration to facilitate germ layer formation and body axis establishment in the developing embryo. And I am pretty sure you might have definitely studied all this in developmental biology classes. Wnt pathway also determines the sex of the developing embryo. So you might have studied about XX and XY. You might have studied if there is XX chromosome, it leads to the formation of female. If it is XY, it forms male. But other than this XX and XY chromosome, the another factor which is very important in the determination of sex of the embryo, that is Wnt pathway. Wnt pathway plays a very critical role in determination of sex of the embryo, whether this is going to develop as male embryo or female embryo or transgender. In those cases, this Wnt pathway plays a very important role. Wnt signaling pathway is also involved in regulation of bone formation. This Wnt pathway has also been shown to be involved in different types of diseases such as type 2 diabetes mellitus, neuronal disorder, rheumatoid arthritis, systematic sclerosis, inflammatory bowel disease, and cardiovascular diseases. So from this slide, you can easily understand that this Wnt signaling pathway has a very important role to play in human physiology and in the development of various diseases in the humans. Now let us have a short quiz, whether you have the memory of previous things that we discussed or you forgot. So first question is, what is the main difference in canonical and non-canonical Wnt pathway? Second question is, ubiquitination of beta-catenin is done by. Third question is, LRP receptor is phosphorylated by. Fourth question is R-spondin LGR5 interaction leads to Wnt signaling. Fifth question is YAPTAZ effect on Wnt signaling is FBXW7 affects Wnt signaling as. So if you have listened the previous discussion very carefully, I'm pretty sure most of the questions will be able to answer. If you are not able to answer some of the questions from this, by the end of this discussion, definitely you will be able to answer those questions as well. Now, let us try to update a little bit about whatever we have discussed about this Wnt signaling pathway. So far we have discussed if there is no Wnt ligand, this destruction complex, which consists of X in GSK3 beta, CK1 alpha, APC and beta TRCP, this is going to destroy β-Catenin by the process of phosphorylation and ubiquitination. GSK3 beta is going to phosphorylate the β-Catenin, beta TRCP is going to ubiquitinate the β-Catenin, and because of this phosphorylation and ubiquitination of β-Catenin, this β-Catenin is going to get destroyed by the proteasome. But other than this, there is another component which we'll try to understand here. So we have a receptor called LGR5, and we have protein called RNF43 and ZNRF3. Now when there is no ligand for this is R-spondin, which you can clearly see here. If there is no R-spondin, in that case this RNF43 and ZNRF3 is going to act on this result, and this result will be removed from here. But when this R-spondin ligand is present, this R-spondin protein will bind to this LGR5 receptor, and interaction of this R-spondin and LGR5 receptor leads to deactivation of RNF43 and ZNRF53. So if there is an interaction between R-spondin and LGR5, RNF43 and ZNRF3 will be deactivated and Frizzle will be able to act on the plasma membrane. So this R-spondin works very much in parallel to this Wnt signaling, and this R-spondin actually positively affect this Wnt signaling. When we say R-spondin positively affect Wnt signaling, what we mean is R-spondin actually induces this Wnt signaling pathway. It supplements the activity of this Wnt signaling pathway by regulation of Frizzle receptor. So if there is high amount of R-spondin, the presence of or expression of this Frizzle receptor on the surface of the membrane will also be high, and this will induce the interaction between Wnt and Frizzle receptor. Another molecule which we can discuss here is this YAPTAZ. This YAPTAZ molecule, which is a component of some other cell signaling pathway, this YAPTAZ also is involved in this destruction complex. So YAPTAZ actually acts as one of the component of this destruction complex, which actually induces the beta-catenin destruction. So YAPTAZ actually helps in inhibition of this Wnt signaling pathway. But in case of presence of this Wnt ligand, this YAPTAZ actually activates this Wnt signaling. So what basically I am trying to emphasize here is this YAPTAZ has a dual role in Wnt signaling pathway. In the absence of Wnt ligand, this YAPTAZ inhibits the Wnt signaling pathway. In the presence of Wnt ligand, this YAPTAZ actually activates the Wnt signaling pathway. So this YAPTAZ is known to have dual role in Wnt signaling. In the absence of Wnt ligand, YAPTAZ acts as the component of destruction complex and inhibits the Wnt signaling pathway. In presence of Wnt ligand, YAPTAZ is free from the destruction complex and it actually activates the Wnt signaling pathway. So it changes its role with the presence or absence of Wnt ligand. In absence of Wnt ligand, YAPTAZ is an inhibitor of Wnt signaling pathway. In the presence of Wnt ligand, YAPTAZ is actually activator of Wnt signaling pathway. That is one important point you should keep in mind. Now, we have already discussed this, that non-canonical Wnt signaling pathway has two types, Wnt PCP type and Wnt calcium type. In case of Wnt planar cell polarity pathway, this DAM1, which is the inhibitor of rho A, is de-inhibited, and as a consequence of that, this rho A is free and activated. This rho A along with this JNK protein actually activates ROCK, and it finally leads to the activation of the target gene. In case of Wnt calcium pathway, Wnt calcium non-canonical pathway, the interaction of Wnt ligand with Frizzle leads to activation of phospholipase C. Phospholipase C activates IP3. IP3 leads to release of calcium from the vesicular structure, because these vesicles are the storehouse of calcium. So this IP3 goes and binds to the IP3 receptor, which are present on the vesicle. As a result of that, this calcium is released, and then this calcium release leads to multiple changes inside the cell, which are this calcium dependent, like the pathways mediated by calcineurin, pathway mediated by CAMK2, and pathway mediated by PKC. All these are going to require the presence of calcium. So because of increase in calcium in the cytoplasm, there is a lot of molecular changes happening downstream of this, and this leads to the activation of target gene. Now this slide is trying to emphasize on how we can utilize this Wnt signaling pathway for different pharmacological purposes. So we have discussed different subtypes of this Wnt signaling pathway, and we also discussed that this Wnt signaling pathway is involved in development of different types of cancer. So the obvious question that might have come to your mind is, if this Wnt signaling pathway is so important or is involved in activation of cancer, then somehow if we inhibit this Wnt signaling pathway, we can suppress those types of cancers. So these are different types of molecules which have been developed to inhibit this Wnt signaling pathway, or different components of Wnt signaling pathway. so that this activated Wnt signaling pathway in the diseased case is controlled. So as you can see, we have tried to suppress this Wnt signaling receptor, which is frizzled by these antibodies, and by using these decoy receptors. We also try to inhibit this beta-catenin CVP300 complex. So these are actually some of the important drug molecules which are under different stages of clinical trial, which are like promising candidate to inhibit this Wnt signaling pathway in different diseases. Here we are trying to understand different components of the Wnt signaling which are involved in the development of different types of cancer. So this figure are trying to explain in percentage. So green color represent pancreatic ductal adenocarcinoma. This one represent breast carcinoma, colon adenocarcinoma, and cutaneous melanoma. And these are the molecules, as we have discussed, the important components of this Wnt signaling pathway. So in various types of cancers, these different components of Wnt signaling pathway are actually dysregulated, and this dysregulation of different components of this Wnt signaling pathway leads to development of different types of cancer. So let us try to summarize whatever we have discussed in the second part. Members of the R-spondin ligand family are known to positively affect the Wnt signaling pathway. R-spondin ligand binds to G protein coupled receptor LGR. In the absence of R-spondin binding, ZNRF3 RNF43 target the frizzled receptor for lysosomal degradation. Binding of R-spondin to LGR46 inhibits the activity of ZNRF3 and RNF43 and leads to the accumulation of frizzled receptor on the cell surface. Being transcriptional target of Wnt signaling, ZNRF3 and RNF43 function as negative feedback regulator in LGR5 positive cells. The interaction of ZNRF3 and RNF43 with the frizzled receptor was found to be dependent on disabled protein. In case of colorectal cancer, pancreatic ductal adenocarcinoma, and endometrial cancer, these types of cancers are having inactivating mutation in RNF43. And that's the reason this R-spondin and LGR, RNF43, ZNRF3 becomes very important in case of cancer, because this R-spondin RNF43 axis are known to be involved in different types of cancer. Now at the level of destruction complex, YAP-TAZ, two transcriptional regulator of Hippo pathway, are identified as Wnt regulator, and we have already discussed that this YAP-TAZ has dual role in Wnt signaling regulation. So in the absence of Wnt ligand, YAP-TAZ are part of destruction complex and recruits beta-TRCP, thereby acting as negative regulator of Wnt signaling. We have already discussed this. However, in an activated pathway state, YAP-TAZ and bound beta-TRCP are displaced from X gene by LRP during sequestration of the destruction complex, and free YAP-TAZ can subsequently act as positive transcriptional effectors of Wnt signaling pathway. So we have already discussed this component that in case of absence of Wnt ligand, YAP-TAZ is inhibitor of Wnt signaling. In the presence of Wnt ligand, YAP-TAZ is the activator of Wnt signaling pathway. So this YAPTAZ plays a dual role in regulation of wind signaling pathway. Now, LTS2 kinase, a repressor of YAPTAZ activity, was found to down-regulate wind signaling by competing with BCL9, a co-activator of TCF-LEF transcriptional complex, which is highly expressed in human tumors. Another important protein called PAF, PCNA associated factor, enhances wind signaling by recruiting histone methyltransferase EZH2 to the TCF transcription complex. So these are some of the important molecules, LTS2 kinase and PAF, been recently discovered, which have been shown to play a very important role in the regulation of wind signaling pathway. Now, wind-dependent stabilization of protein, wind stop, has been introduced as a novel beta-catenin dependent mechanism. Canonical wind signaling, which peaks during G2M phase due to priming of LRP6 by cyclin Y-CDK, was shown to promote beta-catenin independent stabilization of proteins. Beta-catenin independent wind cascade leads to inhibition of GSK3 beta and the subsequent blockade of polyphosphorylation and polyubiquitination of target proteins. Targets include prominent oncogenes such as CMYC, which is degraded by the E3 ubiquitin ligase FBXW. Loss of APC is the main driver of wind signaling colorectal cancer. Loss of basal wind stop leads to increased microtubule assembly rates and subsequently chromosome instability. So this wind stop has very important role if we talk of chromosomal instability in colorectal cancer cells. So that's all for the today's discussion in wind signaling pathway. Thank you for your time and patience. And if you like the video, you should subscribe and share these videos as well. Thank you all.